In this video, we'll be looking at organismal development of two particular organisms, prokaryotes and protists. Basically, what we're going to do is we go through development of organisms. We're going to be looking at how they reproduce and if there are any developmental things that are of importance. The main developmental issues we'll be looking at are for animals, including ourselves. So, review quickly. The difference between sexual reproduction and asexual reproduction has to do with how the DNA looks afterwards, I guess, if you want to think of it that way. With asexual reproduction, uh, there is an offspring created, and that offspring is an exact clone of the parent. There's basically mitosis is happening. There may be a fancy a word for that, or it may be a lesser form of mitosis, but essentially mitosis is going on to create a new organism. Sexual reproduction has to do with the production of gametes, both sperm and egg, mostly. It's just two different gametes, and those gametes combine to form a zygote. The gametes are haploid in nature, and the zygote is always diploid in nature. And so again, the idea or the difference between asexual and sexual reproduction, which we talk about all the time, asexual reproduction is fast. There's not a lot of variation. Sexual reproduction involves the selection of a mate, or at least finding other organisms, um, because some don't necessarily need a particular kind of mate. But there's a lot more variation in sexual reproduction, which we see that is the most successful form of reproduction, just looking at evolutionary history. So looking at then a couple of different forms of asexual reproduction. One of them is called binary fission. Binary fission we will hit on just briefly here. I'll mention again in a moment. Binary fission is just simply one cell becoming two. Bacteria are a good example of this. Budding is another example of how organisms can asexually reproduce. Budding is basically where a polyp like this from jellyfish or some other structure basically generates a little structure that ends up kind of, you can see here these buds, and they almost like a tree branch. That little branch could break off, form another organism, or it could just grow there as a colonial kind of organism. Nonetheless, that bud creates a identical clone. And lastly is fragmentation or regeneration. Seen here in flatworms or Platyhelminthes, also seen in starfish. Basically, part of an organism breaks off. That part that breaks off can grow a whole new organism. The part that is broken off can be replaced by the other organism. It's essentially a way to create organisms by being broken in half. This happens to, for starfish in particular, it's a protection against predation. A portion of them gets eaten, they can regenerate that portion, and it's helpful. So let's look at bacteria in particular. I mentioned again, binary fission is how they reproduce. Binary fission is very stripped down cell division. 3 steps. The cell grows, the nucleus divides, or the nucleus, or there's no nucleus. The DNA replicates, and then these 2 cells split into cytokinesis. Very simple sort of thing. Again, the idea, exact clones created. Very simple. And so we're going to look at 3 ways that there can be variation in bacteria, because of course binary fission doesn't create any variation. One of those ways is called transformation. You can see here from the picture. This is essentially a bacteria taking up DNA from an outside source. A lot of times this is artificial, meaning that we do this. We cause a bacteria to learn a new trick, so to speak, to be able to make a new protein that it wasn't able to do before. We are able to recreate lots of our own hormones by doing this, and so this is an important part of our own medical advance. Another picture, or another idea here, is bacterial transduction, and this specifically has to do with the bacteria interacting with a virus and that virus causing the DNA of the bacteria to be changed. Could be negative, could be a positive change, depending on obviously if the bacteria is able to live through that or reproduce. And so some bacteria over time have been changed by viral DNA, and it's caused them to be changed either for better or worse as far as that particular species is concerned. And then lastly is conjugation. Conjugation is the idea of bacteria swapping DNA. It's the best way of saying this. You have one bacteria that has something called an F factor. The F factor allows them to produce this structure, which is sometimes called a conjugation tube. Here in this picture, you can see it called a sex pilus. And through this tube, they're able to exchange DNA. Again, this is not sexual reproduction because there's no offspring created. But what is happening is these two bacteria are exchanging DNA, and so it allows variation to enter into their populations. And so next, let's look at protists. Protists are basically eukaryotic organisms that are not plants, animals, or fungi. It's a very loose kind of categorization of them. They are, many of them are sexually reproducing, many of them are asexually reproducing, some do both. You see in the picture in the upper left here, you see this bacteria or this protist is undergoing cell division, very simple kind of cell division again. Some of these organisms can produce sexually, and they do so simply by, again, creating gametes. Those gametes combine and they form a new organism. It could be that the organism produces both male and female gametes. It could be that they have to find another organism. There could, meaning that there are two sexes in protists, but by and large, they are organisms that are able to reproduce both sexually and asexually.